Hello.
Hey, Jackie, how's it going?
Good, how are you?
Good, good.
I'm a participant in Brian's study. So I'm trying out your 24-hour BP.
Oh my goodness. Are you gonna use it during the sleep?
Yeah, this is my third day.
Oh my gosh.
Have you tried it?
Yes, yes, several times. Do you have it set up that it goes off every 30 minutes or
During the day and then every hour at night.
at night? Okay. Yeah, you definitely wake up with. It's not like you can.
Yeah, the first night I did, the second night wasn't so bad. So
Yeah, it gets better over time, but yeah, it's
yeah,
not
the first night was rough.
good. Okay, I'm glad it's being used. That's for sure.
Yeah, it's actually pretty cool. Thanks for doing that. Thanks for doing this today.
Yes, absolutely. Maybe offline we can talk about more about the teaching and all that.
Yeah.
I just seen some emails and I've been out of the loop. So let's.
No, I just walked. Yeah, I just walked by his office earlier this morning, and he said something. So he said,
Okay.
"Send me an email to remind me of the conversation." So
Okay. So let's talk offline just to make sure we're all on the same page.
yeah, no, that sounds good. And Dean's working on the nerves. He thinks they'll be done tomorrow. So hopefully done tomorrow.
Great. Fantastic. And I think we should have the insulin data. The assay was completed. I just haven't I haven't seen the data yet. So.
Oh, if they send it to me, I can just add it to the master thing.
Yeah, I asked the student Salvador to send the data. He hasn't. He's probably still working on making the calculations. I don't know.
Okay.
I will remind him again. He did the assay on, I think on Tuesday or something like that.
All right.
So, yeah. And I think tomorrow, I think Gavin and Rogeri are doing some additional experiments to increase the sample size of those aortas. So,
On the beta ones, the panel C.
yes, we had to increase the ends there, and I think in another condition we only had 6, and we were gonna increase the end to 8 at least, something like that. So we need to boost those ends a bit more, but otherwise I think it's in pretty good shape.
Yeah, I agree. That looks pretty good. Let's see.
So after this I have another meeting. Do you want to chat more with about teaching next, like tomorrow or something like that? Do you have time to chat more about that? Or
Yeah, I do. Let me look. I probably have most. Oh, I teach in the morning, but my whole afternoon's open, so I can do any time in the afternoon. Or I teach till 11. I could do it at 11 till noon.
yes, I have times tomorrow too. So let's touch base. Maybe have a Zoom or something. I don't know if Chris needs to be involved or not, or you and I just have to come up with a plan. Whatever.
Yeah. I can send you something. Right now.
Hey Jill, when are you practicing those biopsies with the turkey?
We're practicing with TIFO on the 14th.
Okay.
And, but we went down yesterday, we're going again tomorrow, where literally we're doing walkthroughs.
Yeah.
How to do this. So when John comes, we're hoping they've got down how to do the sterile procedure. They're comfortable. They just basically know what John is going to tell them to do.
And the video that I sent you, the Jove video, they got from, I think they did from Appalachian State University.
Yeah, it was really good. And
it's really good.
That Ying went through it and almost literally wrote word to word and wrote an SOP.
Cool.
Out of it. And we walked through the SOP yesterday and, corrected it and that type thing. So,
okay.
And so now we're really at the point of just practicing. We've practiced with meat and really just being with John. And so he'll be here just in the morning on the Monday, and we hope to get 3 people done.
Mm-hmm.
So 3 biopsies. So
If you
We're
do
moving forward.
3 biopsies per leg, that's 6 biopsies per subject, that's 18 biopsies. I'm just kidding.
Yeah, wouldn't that be great?
Jill, is this
Yeah.
a quality improvement project? Did they let you go under
Yes,
that?
it is.
Excellent.
Yep.
Excellent.
Yep. And so this gets us going. And then Elizabeth, we're hoping that you can, fill in so we can practice a little bit more, and we're looking at probably Tuesday the 15th for that one day, because that will give us a bunch right in a row.
And I think that's the day I told you I was free.
Yes, it was.
And were you able to find all the supplies that I had at the CRC that are not expired?
I found your supplies, I found Elizabeth's, and then we ordered,
More.
I took the list that Lauren gave me and we ordered, well, I told him to order some stuff, he ordered everything on it, so we got plenty of everything.
Okay, just make sure you go through the stuff that we have that is close to getting expired.
Yeah, I'll go through that
Feel free
again.
to throw away, but
Yeah.
we were pretty loaded with things, and we completed the study, and we had a ton of stuff still left. So
Yeah, that's in that box, right?
yeah, it's in the box, but also on the shelves underneath.
Oh, I didn't go through the shelf. I just went through the
Yeah.
box.
Go ahead. The shelves they're labeled Padilla, and to just grab stuff.
Yep. Sounds good.
Is someone slated to introduce our speaker? Who is that?
Yes, it's going to be me.
Oh, wonderful.
But I don't see our
Speaker.
celebrity. I don't see our celebrity vendor here.
Celebrity.
There he is.
Ah, there he is.
Yay.
Oh, here he is. Okay.
I had something that ran right up to this. I wanted to make sure I got here.
You're waiting to be called a celebrity.
Right. I don't know about that.
I'm not joining until I'm considered a celebrity.
Yeah.
You are considered a celebrity.
Dr. Lindbergh, do you need to give some initial introduction, or is it all me?
I'll just do a quick one that this is the last seminar of our fall seminar series. We'll resume again in the spring, but that's it. Just to let everybody know, this is the last week, so seminar next week.
Can I jump in and make one reminder announcement for all the students that are registered for seminar? I did send out an email to please email me your seminar notes. Just a reminder. Thanks.
Okay. So good afternoon, everyone, and welcome to our NEP seminar series. My name is Jaume Padilla, and I'm a faculty member in nutrition and exercise physiology. Today is my privilege to introduce a colleague and a friend, Dr. Shawn Bender, who is an associate professor in the Department of Biomedical Sciences. Dr. Bender obtained his PhD in biological sciences at Ohio University, and thereafter he moved to Mizzou for his postdoctoral fellowship under the mentorship of Dr. Harold Laughlin in the Department of Biomedical Sciences. It was during that time that Shawn and I overlapped as a postdoctoral fellows in the same lab, along with others. And we definitely have some really great memories about this time. In 2011, Shawn was promoted to research assistant professor in the Department of Medicine. And in 2014, he came back to the Department of Biomedical Sciences as an assistant professor on tenure track. This year, Dr. Bender was promoted to associate professor with tenure. In addition to his faculty appointment at MU, Dr. Bender is also a VA investigator. Dr. Shawn Bender has been very active within the Biological Society and the Microcirculatory Society, serving on multiple committees. He's also very active as a reviewer for the American Heart Association. Dr. Bender's research is focused on elucidating the mechanisms of coronary, vascular, and cardiac dysfunction associated with obesity and insulin resistance. And his work is currently funded by NIH R01 on this topic. And this is also the topic of today's talk. Thank you, Shawn, for taking the time to meet with our students today and also taking the time to present your research. We very much look forward to it, and the floor is yours.
All right. So it says my screen sharing is paused. Can you guys see the PowerPoint?
Yes. Can you put it on?
Yep.
Make sure that it's displayed correct. Yes. Looks good.
Okay, does that look? Let me flip my settings. Swap. There we go. All right, can you guys see the full one, or are you seeing presenter view right now?
We're seeing on presenting mode.
Of course. My monitors always put these things backwards. All right, let me put it back.
Should we see the PowerPoint now, or what should we see?
Okay, so do you see the title slide now?
Yes.
Okay, good. All right. Well, thanks for that, Jame. And I had a great time talking with the students at lunchtime today. That was a lot of fun. And it brings back memories of things like my postdoc that Jame mentioned, but also grad school. And it's always a good time to reflect on where I've come from and how grateful I am actually to still be at MU. So so today I'm going to talk about what my lab's been working on for the last probably 5 or 6 years in related to the mineralocorticoid receptor. And this question that I asked in the title, is there a vascular origin of cardiac dysfunction and obesity? So I hope that in this talk, I'm able to make a case, and that would be my goal. And I suppose maybe even to convince you that coronary microvascular dysfunction is a significant contributor to cardiac dysfunction and obesity and metabolic syndrome. And I have primary there, maybe it's even a primary contributor to cardiac dysfunction and obesity and metabolic syndrome. I'm sure that it goes without saying to a group in a Department of Nutrition that we have an obesity problem in the United States. This GIF here is showing the percent of the adult population that is self-reported as obese from 2011 to 2019, and you can appreciate that as we go from 2011 to
Shawn, for some reason we're not seeing the next slide, so we're still on the first.
Really?
On the first slide.
He might have to reload.
That happened to me one time I was teaching.
Okay, so what do you mean, Elizabeth? Reload? Exit out of screen share?
Yeah.
Okay, stop share.
Just get back in.
I think it's better if you share your desktop. I think that's what
monitor.
Dane gave me that secret one time as I was teaching. You have
so you share first and then you go to PowerPoint, you mean?
Yes. Now we are on your
So you're seeing the
overarching aim.
Okay, so now you see the map. Okay.
Yep.
All right. Okay, so all right. Good. Thanks for calling that out. And please, if you have questions during the talk, please don't hesitate to jump in and ask them. So so this map, I'm sure, is something familiar to all of you. And as the colors get warmer, so do the bodies involved. They certainly are much more well insulated. And so I don't think I have to go into any more detail with this slide. But from a cardiovascular perspective, I think it's really important that we appreciate the ramifications of this more broadly. And so if you think about it, somebody, especially with childhood obesity becoming a greater problem, if you're overweight and obese at age 18, statistics from over a decade ago estimated you had a 50 to 75% risk in your lifetime of developing diabetes. And then if you are obese and diabetic, then you have an 80 to 90% lifetime risk of cardiovascular disease. So I don't think that there's any, there's no surprise among the audience here that cardiovascular disease is the #1 cause of death in diabetics. And I should say, since this is a nutrition group, the data that I'll present comes from 4 different models, research models of obesity. They're not all necessarily the same as far as the metabolic derangements that are associated with that obesity. And so that's why my title focused on obesity, and here drawing the relationship between obesity and the ultimate development of diabetes and the role that then plays in the development of cardiovascular disease. So one prominent dysfunction that's associated with diabetes and obesity in the coronary circulation that I'm most interested in is the dysfunction of the coronary microcirculation. And so clinically, that's measured by one parameter, which is called coronary flow reserve. And that's basically the ratio of baseline coronary flow to maximal hyperemic flow induced by some agent like adenosine, for instance, to induce maximal vasodilation of the coronary circulation. And we've known as far back as the mid-90s and even earlier on some studies that diabetes induces impairment of coronary flow reserve, which indicates a dysfunction of the coronary microcirculation. And the clinical literature has done people like me that are interested in the coronary microcirculation a lot of favors in the past 10 or 15 years with some really nice clinical studies showing the importance of coronary microvascular dysfunction to ultimate cardiac mortality. And so this study that was published in 2012, and here I'd ask you to focus on the red bar and the blue bar. And what they're showing you here is annualized cardiac mortality. This study followed patients out for 3 years. And the two groups I want you to focus on, the red group is a group that has diabetes, no atherosclerotic coronary disease. So these are unremarkable on coronary angiogram, and they have diabetes, and they have a coronary flow reserve below the median value, which would be indicative of coronary microvascular dysfunction. The blue group is the group that has diabetes, no epicardial coronary disease, and a preservation of coronary microvascular function. And so you can appreciate that the cardiac mortality, you could make a case here that the preservation of coronary microvascular dysfunction is strongly associated with an overall prevention of cardiac mortality over the period evaluated in this study, to the extent that the group that had preserved coronary function with diabetes had a mortality similar to patients that didn't have diabetes, didn't have atherosclerosis, and didn't have any evidence of myocardial perfusion defects in this study. So the preservation of microvascular function, you could extend this study to say it abrogates cardiac mortality, when in reality this is an associative clinical study. And to take this one step farther to make a case for the importance of coronary microvascular dysfunction, a more recent study came out and evaluated what is the association to outcomes in patients that have coronary microvascular dysfunction, diastolic dysfunction, or the combination of the two. And early studies of the coronary circulation showed that if you impair coronary blood flow to the heart, the earliest defects that you see in response to that are diastolic defects, defects in the heart's ability to relax. And that is one of the earlier defects that shows up in obesity in patients as they progress into cardiac dysfunction with obesity. And so in this study, they looked at a much broader population, not just diabetics, and looked at the adjusted annualized rate of, this is heart failure with preserved ejection fraction, which is a much more prominent form of heart failure in obese patients that we currently actually don't have treatments for, or good treatments for. And you can, if you go from left to right here, what they're showing you is the annualized rate of HFpEF hospitalization actually in normal patients. Then the red here are patients that have diastolic dysfunction alone. These are then the checkered blue are patients that have coronary microvascular dysfunction alone. And the red checkered here are patients that have both diastolic dysfunction and coronary microvascular dysfunction together. And so the take-home point of this slide and this study was that patients that have both dysfunction of the coronary microcirculation and an impairment in diastolic function have a 5-fold increased risk of heart failure with preserved ejection fraction, or incident heart failure with preserved ejection fraction. So I hope that at this point, I've made a strong case that the coronary microcirculation is important, and that the dysfunction of the circulation has ultimate relevance to outcomes in patients that are unfortunate enough to have it. And so my lab, as it relates to mechanisms of microvascular dysfunction and obesity, has spent the last 5 or 6 years focusing on the mineralocorticoid receptor. And so the question initially would be, well, why focus on the mineralocorticoid receptor? So for the students, and we were all students at one point, so I guess I'm speaking to everybody. For the students in this room, you've learned about the renin-angiotensin-aldosterone system. This is a schematic here from my graduate mentor's website that neatly lays out how this works. And you've learned renin and Ang2, and then this little guy over here in the corner that gets hidden in some of these schematics and doesn't probably get as much attention as it should, aldosterone. We know much about it, and you probably only learned about it in class for the role that it plays in salt and fluid balance in the kidney through activating the mineralocorticoid receptor. So certainly the kidney is heavily invested with mineralocorticoid receptor, and that's where we know a lot about it. But I'll go on in the next slide to tell you that that's not all that we know. But first, I'll make the point to define some things here, that there are clinical antagonists of the mineralocorticoid receptor, spironolactone being the most common first generation MR blocker, eplerenone, a second generation, and the most recent finerenone, which is still going through clinical trials, is a non-steroidal blocker of the mineralocorticoid receptor. And these have been somewhat frontline therapy, certainly for the treatment of hypertension for many years, and for heart failure with reduced ejection fraction, because the studies, the Ephesus and Rails trials in the early 1990s showed a dramatic benefit of MR blockade in reducing mortality in patients that have HFrEF. And so we're interested in how the MR may contribute to the coronary dysfunction and cardiac dysfunction and obesity and diastolic cardiac impairments. And that is largely predicated on the well-established paradigm that RAS activation is common in cardiometabolic disease states. And now we know much more about the mineralocorticoid receptor. So in the last 20 years, there have been a number of studies showing that it's much more ubiquitously expressed than you might imagine from your physiology textbook. It's expressed in the vascular wall, and that's where we spend our time being interested in its functions. And it plays a role in cell proliferation and migration and oxidative stress, a very well-defined mechanism of MR-dependent signaling. It's involved in fibrosis and remodeling, and ultimately has been shown to contribute to vascular aging and obviously hypertension and stroke and NMI. And so we asked the question, how could this be involved, or why could this, in what ways might this be involved in coronary dysfunction specifically? My interest in this was piqued years ago in reading a review in the Journal of Hypertension by John Funder and Morag Young, who trained with him. And they make this case, this statement that I quote here, that detailed time course studies in RAS models of experimental hypertension and end organ damage have shown that the earliest discernible events appear to include an intense coronary vasculitis. So early studies pointed to coronary dysfunction being one of the earliest effects of an inappropriate activation of the RAS in these models of, in this case, hypertension and organ damage. And about the time that I was finishing my postdoc, or getting close to finishing my postdoc with Dr. Laughlin, a group at Harvard published one study, and they published a second one since then, showing that MR blockade does indeed improve coronary flow reserve in diabetic patients. And so what you're seeing here on the left is the myocardial perfusion reserve, which is analogous to coronary flow reserve. This was a randomized crossover design in patients after treatment with hydrochlorothiazide to account for the blood pressure lowering effect of spironolactone, or with spironolactone. And on the right, you're seeing a separate study using this is a eplerenone on the left, spironolactone on the right. And this is the change in coronary flow reserve. You can appreciate that coronary flow reserve goes up with spironolactone, but not with hydrochlorothiazide and not with placebo. And so these studies make a case that there's a blood pressure independent effect of MR blockade to improve coronary flow reserve and coronary microvascular dysfunction in patients with diabetes. And so this piqued my interest and really is what got me going down the road of how might the MR be involved in coronary microvascular dysfunction in obesity and diabetes. And so the first study I did was with the Zucker obese rat. I actually inherited these rats from Bill Fay. He was about one of those emails on campus, hey, we have these rats that we ended up not needing. Does anybody want them? And so we got these rats and treated them with spironolactone for 21 days, beginning at 29 weeks of age. And we designed this study to be very specific that we would have a treatment design. We knew from previous work that by 29 weeks of age, the Zucker obese rat has both coronary microvascular dysfunction and cardiac diastolic dysfunction. And so we started the treatment at 29 weeks of age in order to see if we could reverse the deleterious effects of obesity and insulin resistance on cardiac and coronary function. And just by way of kind of showing all of the descriptive data here, we published this paper in 2015. The spironolactone had no effect on blood pressure. It had no effect on cardiac hypertrophy. The metabolic parameters were unchanged. And the kidney dysfunction also the kidney dysfunction associated with obesity in this model was not impacted by this dose of spironolactone. So you can appreciate that proteinuria, the dramatic proteinuria that these rats have, was not reduced by spironolactone. But what was changed, thankfully for me, was that coronary microvascular dysfunction was improved. So, or prevented, or treated, actually, in this case. It's as I mentioned, we treated these rats. And so what you're seeing here are is vasodilation to isolated coronary arterioles from these animals. And you can appreciate that the open circles are the Zucker obese animals had a decrease in dilation to both acetylcholine and insulin. And that was treated or reversed by treatment of these animals with spironolactone. And since Jame's here and he likes insulin, and he's actually on this paper, so I know he knows this, we also showed that AKT phosphorylation induced by insulin was improved by treatment with spironolactone in the Zucker obese rats, making a case that vascular insulin resistance is improved by spironolactone in this model of obesity and insulin resistance. And to our surprise, is at this point in 5 years ago, it was actually quite unclear whether there'd be a benefit of Spiro on cardiac dysfunction and obesity. Echocardiography revealed that impairments in diastolic wall motion assessed by E prime A prime in the obese animals was prevented or treated by spironolactone, as well as the propagation velocity of mitral inflow. Both diastolic parameters were improved by treatment of these obese animals with spironolactone. And this was associated with reductions in cardiac oxidative stress and cardiac fibrosis. And so there were kind of systematic effects here within the heart on both the coronary circulation and the cardiac function itself, along with oxidative stress and fibrosis. And we took this study a bit further to help us be more careful about what could potentially be contributing to the improvement with spironolactone. And actually much of the data on this slide was generated by Jame and Nathan Jenkins, who was a postdoc with us in Dr. Laughlin's lab. And we show in this paper that not only was the cardiac and coronary dysfunction prevented by spironolactone in obese animals, but so too was the adipose inflammation in these animals, along with some reduction in circulating cytokines, predominantly MCP1. Surprisingly, circulating oxidative stress was not reduced in T-bars, and a measure of circulating oxidative stress wasn't changed. So Spiro treatment then in this model attenuated many aspects of the inflammatory and cardiovascular phenotype of obesity in this rat model. And so this really set the stage then for further exploration of how the MR could be contributing to the coronary and the cardiac defects associated with obesity. And one of the questions that I wanted to ask, well, there were two questions actually that I wanted to ask next. One, could we repeat this in another model of obesity, or a model that had even more severe cardiometabolic derangements? And two, what was the association of MR signaling to transcriptomic changes in the vasculature? Because the MR is a steroid receptor, and presumably much of the chronic effects of MR activation are occurring through changes in the cellular transcriptome. And so we utilized a model to answer these next questions that I had used as a postdoc in Dr. Laughlin's lab, the OLEF rat, which I know many of you on this Zoom are familiar with. This is a rat that has a CCK mutation in the brain. It's hyperphagic, gets dramatically obese over time eventually. And this is data from one of Scott Rector's papers showing that over time, insulin levels fall and they do progress into type 2 diabetes from being initially insulin resistant or pre-diabetic. And so this is a unique model in that sense, unlike the Zucker obese rat that seems to stay insulin resistant forever and has high elevated insulins forever. This model actually will progress further into a diabetic state with reduced insulin levels. And so we took advantage of this model to answer these two questions. And the data, we published this 2 years ago, show very similar results in the coronary function data to the Zucker obese rat. So the acetylcholine-induced dilation was reduced in the OLEF rats and prevented by spironolactone, or treated actually, I should say. I'll back up. we also designed this study to be a treatment study. So we knew the first figure of this paper is establishing the timeline of coronary dysfunction. And so we knew at the age of 32, at the age of 32 weeks that we did these experiments at, that there was established coronary dysfunction, and we then intervened to try to reverse it. And so the acetylcholine-induced dilation was reversed. The insulin-induced dilation that was impaired in the OLEF rats was reversed, and there was no change here in the obese animals with smooth muscle-dependent dilation to nitric oxide. And we also went to answer a question we hadn't answered in the Zucker study, which was what's the impact of MR blockade on the constrictor side of coronary function? So the OLEF rats exhibit an increased constrictor response to both endothelin and U46619. And surprisingly, spironolactone had very little impact on the obesity-associated or diabetes-associated hypercontractility of the smooth muscle cells in the coronary vessels. It did tend to reduce constriction to the L-type calcium channel activator BK8644, but this was modest at best at one dose here. So, like the Zucker rats, the OLEF rats confirmed that Spiro is able to reverse or treat coronary dysfunction associated with diabetes. And this was also associated with reduced cardiac and adipose inflammation, as well as cardiac fibrotic gene expression in the OLEF rats when we did PCR at principal markers of fibrosis and inflammation. And as I mentioned, we also had a question at the time of, well, what's happening to the transcriptome? What could the steroid receptor that acts as a transcription factor be contributing to changes in the cellular transcriptome? And so we took aortas from these animals, certainly not ideal necessarily because we're interested in the coronaries, but we come up against some tissue limitations there at the time. And actually we had some other issues I won't get into, but we ran bulk RNA sequencing on the aortas of these animals and found that there were 157 differentially expressed genes by spironolactone relative to untreated obese animals. And I won't get into this in detail so much as to say that one of the things that came out of this was that the MR-dependent vascular transcriptome that these data defined was quite unique. There were only 4 genes that were differentially changed by blocking the MR in obesity that overlapped with any of the other differentially expressed genes in the literature at the time related to the MR. And so I always make this point in talks, and I think it's an important one to make, that this argues that MR signaling is very context dependent. And so what we see in obesity may not overlap with what we see in, say, isolated hypertensives and in other cardiovascular risk states. And when we dug a little bit deeper into the transcriptomic data, we, with ingenuity pathway analysis, we found that there were enrichments of genes associated with angiogenesis, migration of endothelial cells, and the differentiation of smooth muscle cells. And that then led to a spin-off project that Scott Brown in my lab has been pursuing for the last couple of years of what's the role of MR signaling in the arteriogenic and angiogenic responses in a model of hindlimb ischemia. And so that's what he's been spending his time working on. But the take-home points from this second study are really, to just reiterate them, is that we showed in 2 different models that blockade of the MR is able to treat coronary microvascular dysfunction. And in this along with cardiac dysfunction in the Zucker rats, we see similar changes in inflammatory and fibrotic markers that are reduced by MR blockade in the OLEF rat, and a very unique transcriptomic signature of MR signaling in the vasculature in the OLEF rat. And so, to summarize, which I just did, we see somewhat predictable effects of MR blockade, reductions in oxidative stress, reductions in fibrosis, and adipose inflammation. And interestingly, in both models, these benefits occurred independent of blood pressure and independent of other cardiovascular risk factors like hyperglycemia and hyperlipidemia. And so coming in out of these studies, then we had several unanswered questions. And this is really what the aim of my current R01 was to answer some of these questions. And the first of those being, okay, we have data in clinical studies that shows that MR blockade improves flow reserve, which is an isolated measurement of coronary function by eliciting maximal dilation with some agonists like adenosine. Does that mean that MR blockade is improving coronary flow control in vivo? Is it improving the really important role of the coronary circulation to match myocardial oxygen supply with myocardial oxygen demand? And so that's one of the unanswered questions that we came out of these studies with. And then the second one was, well, does vascular MR signaling contribute to, in any other way, outside of flow control, to changes in the cardiac microenvironment with obesity? And so these are two of the primary aims of our ongoing work and my current grant. And the first question we are tackling in conjunction with a longtime collaborator of mine, Jonathan Toon, who was at Indiana University until this past March, and now currently at the University of North Texas in Fort Worth. And we're tackling these studies with the Ostwald swine model of obesity and metabolic syndrome. And so the way that we're going about these studies is we're utilizing the Ostwald swine that we get here on campus from the swine center, and we're feeding them a high-fat, high-fructose diet for 16 weeks, beginning at 6 months of age. So we're working on sexually mature, almost said rats, pigs here. And we have 3 groups in this study. So we have a lean group that's fed a standard chow, the obese group, and then an obese group that's treated with spironolactone once a day, 25 milligrams, beginning when the high-fat diet is initiated. So this is not a treatment design, this is a prevention design. And the spironolactone is given in a marshmallow. And then all of the other animals get a marshmallow every day as well. And you'd be surprised how reticent pigs are initially to eat a marshmallow. I thought they would eat everything. And the way that we evaluated coronary function in this study was to elicit coronary dilation by graded hypoxemia. And we did this by bleeding nitrogen into the inspired air to lower coronary oxygen supply, coronary oxygen or arterial oxygen saturation, and then coronary oxygen delivery to the heart. And I should say that this study and the one that I'll show after this are both studies that we're currently writing up, and so they're currently unpublished. And the basic hemodynamics of these animals are shown in these panels on the right. The obese animals do have an elevation in blood pressure, a slight elevation in blood pressure initiated by the diet, and compensatory, presumably baroreflex-mediated reduction in heart rate in the obese animals. And spironolactone modestly reduced blood pressure with no discernible impact on heart rate. And so because there's changes in blood pressure here, then we evaluated coronary function both as the absolute changes in coronary blood flow in response to hypoxemia, but also by calculating coronary vascular resistance to account for any changes in blood pressure between groups and between animals. And so what we found initially, not entirely surprising, it confirms why we chose this model in the first place, that the obese animals have a baseline reduction in coronary flow, and that's associated with a dramatic increase in coronary resistance at rest. And surprisingly, obesity did not appear to impair hypoxemia-induced vasodilation. So it was unclear in the literature before we did these studies whether or not hypoxemia-mediated vasodilation was impacted by obesity, and our data say a strong no to that. So this method of dilation in the coronary circulation is not impacted by obesity, despite the dramatic increase in coronary resistance at baseline by obesity. But MR blockade, giving these piggies spironolactone marshmallows every day for 4 months, did prevent the increased baseline resistance of the coronary circulation, and again had no impact on the extent of hypoxemia-induced vasodilation. And so this was an exciting finding for us, kind of validating some of what we'd seen in rodent models to a large animal model. But one of the intriguing parts of this was that despite the improvement or the prevention of the increased baseline coronary resistance by spironolactone in obesity, the MR blockade did not prevent an increase in cardiac lactate production in obesity. And so for those that aren't familiar with cardiac metabolism, the heart is primarily aerobic. And when there is a shift towards anaerobic metabolism, then you have cardiac production of lactate. Normally the heart will take up lactate. So you will see the take up of lactate from the arterial to the venous side of the circulation across the heart. The obese animals have a reduction in that lactate uptake, indicative of some production of lactate, and that's not improved by spironolactone in these animals. So that was an interesting finding, that despite the improvement in baseline resistance, there didn't appear to be much of a shift in cardiac metabolism and the balance of oxygen supply and demand. And the other part of this study that I won't spend a whole lot of time on, but it was one of the focuses of my grant, is whether or not MR blockade improves function of voltage-gated potassium channels in the coronary circulation. So we know that the voltage-gated potassium channel is a primary central regulator of coronary flow control in the heart. And so the benefit of MR blockade here to improve baseline resistance did not involve prevention of dysfunction of KB channels in the coronary circulation. And you can see that here, that if we have the lean animals and we infuse 4-aminopyridine into the coronary circulation, you see a dramatic vasoconstriction that is not present in the obese animals if you infuse 4-aminopyridine, suggesting dysfunction of KB channels. And the spironolactone treated animals also have no recovery of the vasoconstrictor effect of 4-aminopyridine, suggesting a maintenance of dysfunction of KB channels in the obese pigs treated with spironolactone. And we confirmed this with isolated wire myography, looking at the constrictor effect of 4-aminopyridine in isolated vessels. You can appreciate that both the obese and the obese spironolactone animals have a dramatic reduction in the acute constrictor response of isolated vessels to 4-aminopyridine. And in collaboration with Doug Bowles in my department, we showed that the smooth muscle cells by patch clamp have a reduction in cellular KB current that can be eliminated by blocking KV1 family channels with the antagonist DPO1. So again, all confirming that there is KB dysfunction in obesity, and that this benefit of improving baseline resistance by spironolactone is not occurring because of an improvement in voltage-gated potassium channel function. So then what could it be? Well, in conjunction with Aaron Trask, a collaborator of mine and friend at Ohio State University and Nationwide Children's Hospital, he did passive biomechanical measurements of isolated arterioles from these pigs. And you can appreciate on the left, the leftward shift in the stress-strain relationship. Now, I know Luis is on this Zoom, so I'll have to be careful how I describe this because I'm setting myself up for questions here. A leftward shift in the stress-strain relationship that is dictated by a reduction in the strain as intraluminal pressure is increased. And so a reduction in strain is indicative of reduction in distensibility or a stiffening of the arterioles. There is not a change in the arteriolar stress across this range of pressures. And if you calculate the incremental modulus of elasticity, we see that the obese arterioles have an increase in this parameter at the higher pressure of 125 millimeters of mercury, which is indicative of changes in collagen stiffness at the higher pressures, consistent with the reduction in distensibility indicated by the reduced strain. And interestingly, spironolactone prevented this. So spironolactone in the obese animals induced a rightward shift of this curve, prevented the reduction in strain, and prevented the increase in the incremental modulus of elasticity. So despite the fact that we don't necessarily have evidence that there's a dramatic an improvement in, say, maybe the oxygen supply-demand balance, these data suggest that the improvement in baseline resistance could be due in part to a prevention of obesity-associated arteriolar stiffening by MR signaling in obesity. And just for the sake of thoroughness, we also measured capillary density. There's a trend for reduced capillary density in obesity that was not improved by spironolactone. Again, arguing that arteriolar stiffening could be a primary contributor to the increase in resistance that we see with obesity dependent on MR signaling, such that we can prevent it with MR blockade. So we come back to our unanswered questions. Well, does MR blockade improve coronary flow control in vivo? I'd like to say yes and no. So it improves resting coronary resistance, doesn't necessarily seem to make a dramatic dent in the metabolic indices of myocardial oxygen supply and demand. And so it may not necessarily improve cardiac oxygen balance despite improving baseline coronary resistance. So if we take all of this together and say, well, we have evidence that endothelial function's improved in rodent models. We have evidence that inflammation is improved systemically when you block the MR. We now have evidence that it doesn't appear that improvements in coronary flow control are the primary mechanism of, are a primary mechanism underlying the impact of MR signaling in the coronary circulation. What else could be going on? And what could be going on in the microenvironment of the heart? And I will say that the reason we focus on this idea of a microenvironment is because almost everything that we've seen in these other studies has been independent of blood pressure. It's been independent of the metabolic indices. And so it really makes an argument in my mind that whatever MR signaling is doing in the coronary circulation and in the heart is largely predicated on what's happening within the organ itself. And so to explore this, we went back to a model that we used for a number of years. Actually, a variation of this model is what I used for my dissertation in graduate school. And this is the diet-induced model of obesity in mice. And we used a diet that's 45% kcals from fat, 35% kcals from sugars, and this is a mix of sucrose and fructose for 16 weeks. And we did this in female mice. And the reason that we focused on female mice is twofold. The first is we published previously with Luis that male mice don't have, fed this diet for 16 weeks, didn't have a dramatic impairment in endothelial function. This was surprising at the time, but we have data since that female mice do have an impairment in endothelial function fed this diet for 16 weeks. So there do appear to be sex differences in the coronary phenotype induced by this Western diet. On the dilator side of the coronary circulation, the constrictor side appears to be more similar in that both males and females experience or demonstrate an increased constrictor response to the thromboxane analog U46619. And so we focus then on females because they have a greater coronary phenotype, maybe a more dramatic coronary phenotype with both defects in dilation and constriction. And we had previously shown, when I was in Jim Sauer's group, that blocking the MR systemically with spironolactone in female mice prevents cardiac diastolic dysfunction in female mice. And so back to the initial slide that I showed you, where patients that have combined coronary microvascular dysfunction and cardiac diastolic dysfunction have a 5-fold increase in ultimate incident heart failure with preserved ejection fraction. This model of obesity and induced coronary and cardiac dysfunction seems to be quite relevant to that patient population. And spironolactone in this study also, similar to what we saw in the rats, attenuated Western diet-induced increases in cardiac oxidative stress and fibrosis, and did have a modest effect to reduce cardiac hypertrophy. We've now gone back and done an additional study with these female mice, and now can show that spironolactone given to these animals prevents the endothelium dependent impairment of vasodilation to acetylcholine, although there didn't appear to be much of an effect to improve endothelium independent vasodilation, despite there being some effect of spironolactone to reduce obesity-associated increases in vasoconstriction. So again, we now are confident that we have a model of both coronary and cardiac dysfunction with translational relevance. And we've applied this model now to a smooth muscle cell specific model of mineralocorticoid receptor deletion. So this is a mouse that we collaborated with Iris Jaffe at Tufts Medical Center for several years. And this is just kind of the confirmation data here showing excised MR in vascular, in the vasculature, as well as in the intestine and the gut, where you have lots of smooth muscle. This is a deletion driven by the smooth muscle actin promoter. And if we look at the aorta, you can appreciate here that in the whole aorta, as well as in the denuded aorta, you have the excised MR confirming deletion or excision of the gene from its location in chromosomes. These mice were crossed initially with a Rosa reporter mouse, and you can appreciate here that the blue staining here showing the locations of the deletion of the MR in the smooth muscle cells. And in young animals, at least, this deletion has no effect on blood pressure. This Nature Medicine paper that this model was first reported in did demonstrate that with aging, this deletion prevents aging-associated hypertension. But at younger ages, there's no effect on blood pressure. And so we've then taken this model and with female mice, feed them the same diet that we've used in the past, and can now say, and this is a paper that we're writing on, that smooth muscle cell MR deletion does prevent coronary dysfunction associated with obesity. So up here on the top are the MR intact animals. Western diet feeding impairs acetylcholine-induced dilation. There is no change in smooth muscle cell dependent dilation to NO, and increases constriction to U46619. And these coronary defects are prevented in the mice that have the deletion of the MR from smooth muscle cells. And after 3 or 4 rounds of these mice, I can say with confidence now that this deletion also prevents the cardiac dysfunction associated with obesity. So what you're looking at here is EE prime, which is a echocardiography-derived estimate of left ventricular filling pressure. An increase in this parameter is indicative of dysfunction. So this is increased with Western diet feeding in the MR intact animals, and that increase is prevented in the knockouts, along with E prime, A prime reductions. This is a diastolic septal wall motion. Reduction in this is dysfunction that's prevented by the knockouts. And this is independent of changes in cardiac hypertrophy. So the obesity-associated hypertrophy is still present in these mice, despite the fact that the heart and diastolic function is preserved by this deletion. And unlike what we see when we treat the animals with an MR blocker systemically, this deletion did not prevent adipose inflammation induced by the Western diet feeding. So you can appreciate that the closed circles and squares here, looking at these markers of adipose inflammation, all remain elevated in the smooth muscle cell MR knockout mouse, again suggesting that what is happening and what could the benefit of MR deletion in smooth muscle cells is on cardiac function is occurring as a function of something inside the heart. It's not necessarily because we're reducing this, what I use as a systemic marker of inflammation in the adipose tissue. And this deletion also didn't change blood pressure or the metabolic derangements associated with obesity in females. So just a couple slides here to wrap up. We really had a roadblock when we got to this point, because there were several questions that were outstanding, trying to figure out how this deletion in this vascular cell within the heart could have these dramatic effects on preventing cardiac dysfunction associated with obesity. We ultimately settled on the best way to approach this would be to evaluate the cardiac cellulome with single-cell RNA sequencing. And we did this in collaboration with Alex Pinto at the Baker Heart and Diabetes Institute in Melbourne, Australia. And he helped us work out the cell isolation technique that he had come up with for adult mouse hearts. And basically it's just a model of grinding up the heart, isolating the non-cardiomyocyte fraction of the heart, flow sorting for living nucleated cells, and then bringing them to the DNA core for single cell RNA library preparation and sequencing. And And then Alex is a master of bioinformatics, and two of his postdocs have been, Malafi and Ian have been working on this with us to do the bioinformatics on this. And so just, 10,000 foot view of what you end up seeing here are a clustering of the non-cardiomyocyte cells based on gene expression by marker genes. And these are the primary marker genes in C for each of these cell populations. And so you can appreciate that we were able to capture immune cells, smooth muscle cells, pericytes, macrophages, fibroblasts, basically every population except the cardiomyocytes. Cardiomyocytes are actually too big to do by themselves. You have to do single nuclear RNA-seq, and so we didn't include the cardiomyocytes. And on the right here is actually the panel of genes that was used to then cluster these cell populations for further analysis. And what I can say at this point, without getting too deep in the weeds on this, because you can imagine that this is just an absolutely massive data set. We've spent the better part of the last 6 to 8 months trying to digest this, and we're now in the process of writing up this paper. But on the top here are the actual cell clusters from each of our groups. So there's no discernible dramatic shifts, say, in the overall abundance of these cell populations with obesity or with our knockout. So this is wild type control, wild type fed a Western diet, the knockouts on control diet, and the knockouts on Western diet. But we do see differential expression of genes. And here we've broken out the cell populations into smaller clusters, because they do cluster depending on how you set up this analysis. We have 9 clusters of endothelial cells that have different gene expression profiles. And you can appreciate that the red here are the number of genes in these clusters that are upregulated in the knockouts by Western diet, and the blue circles here are the number of genes in each of these populations that are upregulated in the wild type animals by Western diet. And the number in the open circle here are the equally expressed genes between the knockouts and the wild types on Western diet. And I show this really, this is the last slide. I show this to make the case that A, there's a lot we're still trying to work through here. But B, I think it's really interesting to look at something like this and see that there are actually several cell populations that there's not much going on in. And so I think this single cell approach really allows you to dissect some of the specificity of the response and some of the more high resolution effects of obesity on these cell populations. And our ongoing analysis at this point, and I could do a whole separate talk on the single cell data. The ongoing analysis at this point points to several things. One is an interesting finding that our Western diet-induced model of cardiac dysfunction, when we do picoseries red staining, we don't see much fibrosis in these female mice. And the single cell data by the gene expression profile in the fibroblasts and the smooth muscle cells backs that up. We don't see an enrichment of fibrosis genes that you might expect in a model of diastolic dysfunction. And it points to a central role of the smooth muscle cell MR in the cardiac inflammatory response elicited by Western diet feeding in these female mice. And so we're dissecting this. We're stay tuned. I think this paper hopefully will be submitted in the next month or two, and we'll get this what ends up being a really massive but really exciting data set. Not only because of the effect of the knockout, but also because it should be the first paper where the cardiac cellulome in the setting of obesity has been described. And hopefully that sets up further studies. And so, and in conclusion, we're able to treat established coronary microvascular and cardiac dysfunction by blocking the MR independent of blood pressure. The Western diet-induced feeding induces a dysfunction in female mice, but not in male mice, that occurs in concert with diastolic dysfunction. MR blockade improves resistance in our swine model and the structural remodeling that's associated with obesity. And the smooth muscle cell MR appears to be a primary contributor to the impaired coronary and cardiac dysfunction in obese females. So I hope, especially with this last study, that I've been able to make a case and maybe convince you that coronary microvascular dysfunction or vascular signaling, vascular cell signaling, is a significant contributor to obesity-associated cardiac dysfunction. And with that, I'll acknowledge all of the people over the last, say, 7 or 8 years that have been contributing to these studies, including folks from your department, but also lots of folks here at MU, and I just noticed I left people off, like Doug Bowles isn't on here, Luis Martinez isn't on here, but I called them out when we showed their data. So there's certainly, these things don't happen in a vacuum, and I'd like to acknowledge our funding sources, and then I'm happy to take any questions.
All right.
I'm clapping for you.
Bender. Very nice presentation. Maybe we can you stop sharing and maybe we can have some time for questions. We have about 7 minutes if anyone has questions.
I'll go first. So I have a question. We do a lot of hypoxia in our lab. So it's fun to see. So why do you think that first that there was an impairment in hypoxic dilation, obesity, and then did you choose another stressor to look at as well, or was hypoxia the only one?
No,
and then why?
Yeah, that's a great question. We actually did both hypoxemia and dobutamine. We're still making our way through the dobutamine data. We're really interested in what happens if we impair supply, and then what happens if we increase demand. And so we did both hypoxemia and then followed that with dobutamine. I think the reason we don't see an impairment in the hypoxemic induced dilation is because I think that dilation is largely dictated by KATP channel opening, and we don't have evidence for this, but I don't think that those channels are necessarily as dysfunctional as, say, the KB channels are in obesity. So I think that's partly why the hypoxemia-induced vasodilation is similar with obesity, and we're still kind of slogging our way through the dobutamine data to make sense of what happens in the alternative scenario where you increase demand rather than reduce supply.
Thanks. Really nice presentation. Thank you for doing this. Thank you for doing lunch today too. I heard it was really great.
That was great. I'm happy to.
Can you tell me about the single cell work, which I think is really exciting? That's a great talk. Kind of what are your challenges aside from data overload? And how, I guess, in terms of the mass of the heart, how do all those cell types kind of color your thinking about the influences on dysfunction and how you're going to go after that?
Yeah, I think yes. So you're correct that there's data overload with the single cell. It's quite, I jokingly said to my lab when we got the data set that I thought that our chronically instrumented pig data was a massive amount of data until we got the 600 gigabytes of sequencing data from the core. We were fortunate from the logistics side of things. We were fortunate that Alex, our collaborator in Melbourne, had just published really what were the first 2 papers in the last couple of years on non-cardiomyocyte isolation in adult mouse heart. So there's quite a bit of single cell RNA sequencing done in the heart of neonates and in the developmental cardiac development field. And so there's quite a bit for younger animals, but he really perfected the isolation on adults, and Chastity in my lab, worked, seemed like unstoppably for about a month and really nailed down that protocol to get those cells. And the other side of it is that the analysis is really over my head. And I'm really thankful that Alex was willing to take that on with us and do that analysis. That it's not a trivial thing, as you can imagine. And I'm hopeful that whatever the reincarnation of our bioinformatics core here at MU ends up being, offers that service to make that a more streamlined process for us here on campus. But I think to your other question, the data sets really forced me to think more broadly about how vascular cells interact with the environment that they're in. I think I came into these studies with a much more coronary flow-centric perspective of the benefit or of the importance of the coronary circulation. And this really forced me, these data together, both the pig study and the mouse study, are really forcing me to think beyond that role of flow control. And I think in this case, our data is making a strong argument that the MR and smooth muscle cells really initiates an inflammatory cascade that impacts the cells locally, but also plays a role in the recruitment of leukocytes into the tissue that then can be a contributor to the dysfunction. So we have other flow cytometry data that corroborates what we're seeing with with the single cell RNA sequencing. And I also think it's exciting because this data set is, in some ways, it challenges this dogmatic way of thinking about diastolic dysfunction, that it's because of a structural stiffening of the heart. We don't see much fibrosis, like I mentioned, and we don't see an enrichment of these fibrotic gene signatures that you might expect with the obesity phenotype. And so I think that's certainly a part of this paper. We'll be trying to push back on that maybe slightly dogmatic way of thinking about diastolic dysfunction as being more or less a structural stiffening, that there are things going on that make the heart actively more stiff, so if you will. And so it's good. It's really stretched me outside of my comfort zone, and it's been a lot of fun.
We have Luis.
Hi, Jaume. Shawn and Jaume. Very, very nice talk as usual, Shawn. Very, very interesting. Lots of questions. One of them is a comment. The first one is going to be a comment just on what you said. I think that something else that we need to think about is that sometimes we just simplify stiffening and fibrosis as well, just by measuring picosurious red.
Sure.
And if there is not, then there is no fibrosis. Well, you know, I'm interested in cross-linking and other types of things that may increase stiffness that maybe are not detected by picosurious red, and you know, we need to consider that. But
yep.
My other question is, do you think it's just the local inflammation that is affecting, for example, the insulin resistance at the level of the endothelium that you see, and how is that reversed by spiral? And what do you think about that effect that you see of vasoconstriction? You saw some vasoconstriction in response to insulin. Do you think it's just a shifting on the nitric oxide versus endothelin-1 pathway, or what do you think is happening there? How is the MR working on the endothelial insulin resistance and the reversal by spiral?
Yeah, so we didn't do insulin in the mouse and the pig studies, but in the rat studies, I think it's a difficult thing to tease out whether the effect of MR blockade is because it's reducing inflammation locally or systemically, because in that study we see reductions in inflammatory markers in the heart itself, but also in the adipose tissue, because that was a systemic blockade. I do think that part of what we see, say in the OLEFs, where you mentioned that we saw a slight constrictor response to insulin, is likely a shift in the nitric oxide endothelin balance in the coronary circulation. So unlike skeletal muscle, for instance, as I showed, you know, insulin dilates the coronary circulation without having to block the constrictor effect of endothelin, right? Which you have to do in skeletal muscle. And so I do think it's a shifting of that relative contribution of nitric oxide to endothelin. And there's also a case to be made. There were studies published about 8 years ago that showed that MR signaling upregulated the hybrid receptor, the IGF-1 insulin receptor, the hybrid of those two receptors, and that the deleterious effects on insulin signaling vascular cells was because of enhanced hybrid receptor signaling and reduced insulin receptor signaling. And you could prevent that by blocking the MR and preventing the upregulation of the hybrid receptor. So I think there's probably an effect of inflammation, whether that's local or systemic. We can't necessarily answer it with our data set. But I do think there's molecular mechanisms at play too, with regard to the balance of insulin receptor and hybrid receptor signaling.
All right. I think there are many other questions that could be asked, but we are running out of time, and so to respect Dr. Bender's time, we'll leave it here. Thank you again, Shawn, for this very nice presentation. I'll definitely have some questions. I'll follow up next time. Good.
Thanks, everybody. It's been a lot of fun.
Thank you so much. And I don't know if Dr. Lindberg has any additional announcements. No.
Nope, that's it. Thank you so
All
much.
right.
That was
Thank you,
great.
everybody. Thank you, Shawn.
Thank you.
Bye.
Say hi to the family.
I will for sure. Same to you.